Hello, good morning. This is Gabriella Heltzel. I'll be covering my selected infectious disease, which is Plasmodium species, commonly known as malaria. So let's just get started. So what is the cause of malaria? Malaria is caused by a protozoan Plasmodium parasite. This parasite can be spread to humans through the bites of infected mosquitoes. There are many different types of Plasmodium parasites, but only 5 can cause malaria in humans. And the most commonly type of malaria parasite and is responsible for most malaria deaths is the Plasmodium falciparum. So we're going to go ahead and look at the staining of a Plasmodium species. Since they are intercellular parasites, one of the best stains for malaria is the Giemsa staining, which is also used for other blood parasites. This is typically used because smears for malaria parasites are stained using the same stains as for blood smears. We're going to go into depth about the microscopic view of the Plasmodium species. You can see in the figure 1 that the trophozoites in Plasmodium falciparum has the characteristic signet ring shape. And also unique to this species is the presence of multiple trophozoites in one cell. As you can see in figure 1, there's shown 3 in one cell, which is unique to this particular species and is not in other Plasmodium species. In figure 2, red blood cells infected by Plasmodium vivax are often larger than uninfected red blood cells. They are approximately 1.5 times the size of a normal cell. In figure 3, this shows the Plasmodium malariae and the characteristic of the trophozoites of Plasmodium malariae showing the ring shape and the tendency of infected cells to be of normal or smaller size. And this is shown and indicated by the arrows. Moving on to the gametocyte, in the Plasmodium falciparum, the gametophytes of this has a crest or banana shape, as shown in figure 4. In figure 5, there's shown the gametocyte of Plasmodium malariae, and this particular one has a round shape about the size of red blood cells, and they have a fine grainy appearance to them. In figure 6, we're shown the gametocyte of the Plasmodium ovale. This particular one has a round gametocyte that is larger than normal red blood cells, and it also has a grainy appearance as well as the Schaffner's dots. Here we're shown a figure of the figure 1 schizont of the Plasmodium malariae. This particular figure shows the schizont containing myriazoites, 6 to 12 giving a coarse, grainy appearance. In figure 2, we're shown the schizont of the Plasmodium vivax. This schizont showing the large number of myriazoites typical of this species. This contains about 16 to 24. Also note the larger size compared to a normal red blood cell. So figure 3 is indicating a red blood cell showing the Schaffner's dots characteristic of cells indicated by the Plasmodium vivax and the Plasmodium ovale. So to the right, I have made a T-chart that kind of goes over the overview of the characteristic differences under the microscope, kind of what the Plasmodium species have and similar to each other and differences, and we kind of went in more depth covering each figure, but this one kind of gives an overview of the similarities and differences. I like to point out the fever in these particular species actually differ as well, not just the appearances, but the symptoms too. So in this slide, we're going to cover the geographic distribution. This current distribution of malaria in the world is shown on the world map to the left, and it kind of shows the pale green, that areas that malaria is not currently affecting, or maybe it was eradicated or never existed. And you can see that the United States is actually currently one of those countries. I did want to put the map to the right to kind of show you that even though malaria is not currently in the United States and not something that we have to deal with, we still have Anopheles species, which is mosquitoes that carry diseases. These can be spread through mosquito contact, and not just malaria, but other diseases that are still in the United States and we still have to deal with. Another thing to keep in mind, travelers are at risk for contracting malaria and is highly variable from country to country, even between areas in the country. So this must be considered in any discussion of any appropriate preventative measures. The highest transmission is found in Africa, south of the Savannah, and in parts of the Oceania, such as New Guinea. In cooler regions, transmission will be less intense and more seasonal, but Plasmodium vivax might be more prevalent there because it's more tolerant of lower temperatures. So mode of transmission, malaria is transmitted normally through the bite of affected mosquito. These mosquitoes are almost always female and of the genus Anopheles, which carry malaria parasites in their saliva glands. The parasites at this part of their life cycle are known as sporozoites, and these are introduced in the host's blood when mosquito takes blood for a meal. From there, the sporozoites, travel to the liver, reproduce. This process takes several weeks, then re-enters the bloodstream. At this point, the patient will begin to experience symptoms. Eventually, the malaria parasites change again into gametocytes, which are picked up by another mosquito, again when it bites the infected person. In this way, the life cycle of the malaria parasite continues, and it's a vicious cycle. Because malaria reproduces in the blood and in the liver, in some cases malaria can be transferred through organ transplant or blood infusion. In addition, malaria parasites can cross the placenta. So this can be transmitted from a mother to her unborn child, either in womb or during childbirth. This is known as congenital malaria. In malaria life cycle, the malaria parasite develops both in humans and in the female Anopheles mosquitoes. The size and genetic complexity of the parasite mean that each infection presents thousands of antigens or proteins to the human immune system. The parasite also changes through several life stages, even when in the human host, presenting different antigens at different stages of its life cycle. Understanding which of these can be useful target for vaccine development, this has been very complicated. In addition to the parasite has developed a series of strategies that allow it to confuse, hide, and misdirect the human immune system. Malaria's infection starts off when an infected female mosquito sits on and feeds off an uninfected person. By doing so, it infects malaria in the form of the sporozoites into circulation of the human being. This is passed on rapidly into the human liver where they get deposited, and then they multiply asexually inside the liver cells and accumulate over the next 2 weeks. At this stage, you will be free of symptoms and will be undetected that you even got affected. Over the next 5 to 16 days, they begin to grow, divide, and yield tens of thousands of haploid cells. These then multiply asexually in the liver cells over the next 7 to 10 days, causing no symptoms. In an animal model, the parasites in the form of merozoites invade the red blood cells and multiply again until the cells burst, and then they invade more red blood cells. This cycle is repeated, causing fever each time parasites break free and invade red blood cells. So why should we study malaria? Did you know the U.S. government is actively spending large amount of money on malaria research? The United States National Institute of Allergy and Infectious Diseases funds the International Centers of Excellence for Malaria Research. This is a global network of independent research centers in malaria countries that support researchers in various aspects of malaria research. This program was actually surprisingly started in 2010. Research activities are being conducted in the following regions, comparing more than 17 countries and over 50 field states. But why is this important to those who live in countries where malaria has been eradicated? Well, the simple answer is that it's for global research and that we are still at risk traveling to other countries. And even though it's been eradicated here, it doesn't put us at a zero risk. The CDC has some fast facts on just how many people are affected by malaria, and the numbers are staggering. Over 3 billion live at risk areas in malaria, and in 2010, malaria caused 216 million clinical episodes and over 655,000 deaths. It's hard to stabilize economics and populations in developing parts of the country and the world with people at risk for such a debilitating disease. Understanding and controlling malaria is very important to many countries' military operations. Service personnel are dispatched to many tropical and subtropical countries where malaria is still prevalent. Sick soldiers are not in the best of interests of the military, and so many armed forces have their own malaria research and prevention groups that work closely with the global research and public health organizations. Roughly 3.2 billion people, almost half the world's population, are at risk for malaria, according to the WHO, the World Health Organization. Although substantial progress has been made in the fight to control and eliminate malaria, the mosquito-borne disease remains a significant public health problem, and this affects us all, even if we live in eradicated countries. So what are the symptoms of malaria? The symptoms of malaria can be seen within 6 to 8 days of the infected mosquito bite. They can include high fever, which can go up to 105 degrees Fahrenheit, profuse sweating, fatigue, nausea, muscle pain, among others. In the long run, malaria can be fatally charged. But what if malaria is left untreated? This could result in anemia, jaundice, mental confusion, kidney failure, a coma, seizures, and even death. Cerebral malaria, which causes the blood vessels of the brain, can result in brain damage. The long-term outlook for patients with these parasites may also be poor. And these patients, malaria may reoccur, and this may cause other complications as well. The system respond to malaria. Plasmodium, the key culprit of malaria, impairs main cells to the immune system. This parasite lives and multiplies in places where immune system can't reach it, which include the red blood cell. When these blood cells get flooded with the parasite, it tends to burst, and this is the time when the immune system gets a chance to attack the plasmodium. And this is what causes the high fever, as the immune response to the attack of the plasmodium on the red blood cells. Here, some of the parasites are successfully destroyed while the other plasmodium that have ruptured out of the red blood cells quickly hide themselves into other red blood cells. This process goes on in the immune system of the patient with malaria when they're only able to develop slow and particular strain immunity. It is the same reason that the patients suffering from malaria often appear to be in good health and asymptomatic. Symptoms of malaria can only appear at regular time frames in some cases. Children are high risk of malaria owing to the fact that the immune system of children are weak and produce antibodies that are not protective. Since the adults have a developed immune system, they are stronger protection against plasmodium. Due to this particular invasion, research has been done to study malaria and kind of see how malaria works in hiding itself and against the immunity of the host. Clinical immunity to malaria is slow to develop and short-lived. One reason is that extensive diversity found in malaria antigens, which facilitate parasite escape from the host's immune detection. Much of the invasion process itself is organized around a key interface that forms between two cells called the tight or moving junction. And this is shown in the figure that shows the malaria and how it bursts the red blood cells and how they're able to detach and avoid the invasion process. However, although each step of invasion has been described in detail under the microscope, they are incompletely understood at the molecular level and only recently described in cellular detail. This experiment contained various combinations of rodent Plasmodium species and inbred mouse strains to be used to mimic human malaria infections. Innate and adaptive immunity are linked as the C cells produced by the cells of the innate system modify the outcome of the adaptation response. This has been important implications for the vaccine design and immunotherapy. In mice, the innate response can limit acute protozoan infections, and with the help of chemotherapeutic agents to facilitate a parasitic cleanse. Surprisingly, this research had actually concluded more questions and more emphasizes the misunderstanding that we have of malaria and how much more research has to be done to quite understand it on a molecular and a cellular level. And I'm looking forward to more research that is being published so I can learn more, because this presentation has made me have more questions than answers.